Thank you for this opportunity, and thank you all for being here. I know it's really late, so I really appreciate it. I'm Sumana. I'm a graduate student in the computational biology group at ETH Zurich, and today I'll be presenting our work on improved pathway reconstruction from RNAi screens by exploiting off-target effects. So just to give you a quick introduction of the existing method and our motivation, a biological pathway is a series of interactions between different molecules in a cell that results in a specific product or change in the cell. So when a host is infected by a pathogen, there are different interactions between the host and the pathogen molecules that are often not well understood. A better understanding of these interactions could enable us to design a suitable drug against the pathogen. And one such way of inferring these interactions is by using observations from perturbation experiments. And RNAi is just one such technology used to perturb the system. Before I proceed further, I'd like to introduce you to some of the common terminologies that I'll be using throughout this talk. So S1 to S4 are called signaling genes, and we're mainly interested in inferring the relations between these genes. E1 to E4 are observed phenotypic effects, and these are associated with the different signaling genes. And K1 to K4 are different knockdown experiments, with K1 designed to knock down S1, K2 designed to knock down S2, so on and so forth. Finally, a network is said to be transitively closed if each node receives edges from all of its ancestors. For instance, here, S1 is upstream of S2 and S3, which are further upstream of S4. So the edge from S1 to S4 makes this network transitively closed. Now, each perturbation experiment yields a different phenotypic effect pattern. So in K1, S1 is perturbed, and since S1 is upstream of all the other genes, all of them are perturbed too in that experiment, and we observe all phenotypic effects. Similarly, in K2, S2 is perturbed, and since S2 is upstream of only S4, we observe E2 and E4. In K3, S3 is perturbed, and we observe E3 and E4. And lastly, in K4, only S4 is perturbed, and we observe only E4. Now, this is the final data set that we get from all perturbation experiments. It should be noted that these experiments are independent and typically performed in parallel. Now, given the data and the knowledge of which gene is knocked down in which experiment, we get a subset relations. That is, the effects observed in K4 are a subset of the effects observed in K2 and K3, which are further a subset of the effects observed in K1. Nested effects models are two-layered graph models that infer signaling networks from such subset relations of phenotype. More formally, a NEM consists of a signal graph phi that encodes the relations between different signaling genes and an effects graph theta that encodes the relations between different phenotypic effects and the signaling genes. The underlying assumption is that each phenotypic effect is associated with at most one signaling gene. In fact, Florian was the one who came up with this class of model. And in their paper, they define a nested effects model, F, as phi times theta. Now, NEMs assume that the perturbation experiments are strictly on target. However, in reality, it has been shown again and again that these siRNAs show strong off-target effects, with the final phenotype being a resultant of combinatorial on-target and off-target perturbations. Thus, under ideal conditions with no off-target effects, NEM successfully infers the true network. However, in this case, in the presence of off-target effect E3 in K2, NEMs likely infer an additional edge between S2 and S3, which is a false positive. An alternative representation of the same setup with an additional layer of known knockdown experiments. The dashed line here just means that K1 was designed to knock down S1 and K2 was designed to knock down S2 and so on and so forth. As I mentioned in the previous slide, under ideal conditions, NEM successfully infers the true network, and in the presence of an off-target effect, it likely infers an additional edge. We propose that by explicitly accounting for an off-target perturbation of S3 in K2, we can successfully infer the true network even when there are off-target effects in the data. That brings us to our model, probabilistic combinatorial nested effects models, or PCNEMs for short. Now, a PCNEM is again parameterized by a signal graph phi and an effects graph theta. However, in addition, we make use of a perturbation map rho that encodes the knockdown probabilities of different signaling genes in each experiment. Now, we can get this information from tools such as Target Scan. And the data now consists of both on-target and off-target effects. In addition to off-target effects, the data is subject to noise with false positives and false negatives. These are modeled by parameters alpha and beta, as in the case of classical NEMs. Now, given n signaling genes, L phenotypic effects, and k number of experiments, and a perturbation map rho with each entry describing the probability of signaling gene S being knocked down in that specific experiment, we compute a propagation matrix pi for a given signaling network. Now, each entry of this matrix is recursively defined as the probability of the signaling gene itself or any of its ancestors being silenced in that particular experiment. We then define PCNEM as propagation matrix pi times effects graph theta. Now, given the perturbation map rho, signal graph phi, effects graph theta, and the error rates alpha and beta, the overall likelihood of a PCNEM factorizes into the conditional probability of each entry of the data given the model parameters across all experiments and all phenotypic effects. Now, the model that we have defined is strictly designed for binary data. So the conditional probability of observing an effect given the model parameters is the weighted sum of false positives and true positives. Similarly, the conditional probability of not observing an effect given the model parameters is the weighted sum of false negatives and true negatives. Now, we do not know the exact topology of the effects graph. So as in the case of classical NEMs, we marginalize across all possible thetas. So assuming a uniform prior, again, the marginalized likelihood reduces to this form. The additional node here just accounts for uninformative effects. Typically, the likelihood landscape for binary data is rugged, increasing the chances of getting stuck in a local maxima. So we developed a novel inference scheme based on adaptive simulated annealing. In addition to inferring the network structure, we can also estimate the error rates. And unlike classical NEMs, we're not restricted to transitively closed networks, and we extend the search space to the entire DAG space. We further proved that PCNEM is identifiable in this DAG space under mild assumptions on the rank of the perturbation map and the range of the error rates. I won't be going into the details of the proof. So now, having defined the model, I will move on to the results of our exhaustive simulation study. So for the simulation study, we first ranked the pathways in KEGG in decreasing order of off-target frequencies, and then sampled networks from the highest scoring pathway. In the first part, in order to compare with classical NEMs, we restricted the networks to transitively closed networks. So we sampled 30 DAGs with high degree of off-target frequencies and simulated data for 3 different pairs of error rates and 4 different number of phenotypic effects. The results are summarized here. So on the x-axis, you have different number of phenotypic effects in the data, and on the y-axis, you have performance being measured in terms of structural Hamming distance, which is essentially the number of edges that are different in your inferred graph from that of the ground truth. Now, each facet corresponds to the performance for a given pair of error rates. And since we developed a new model and a new inference scheme, we first compared NEMs with the existing module network algorithm and NEMs with our algorithm, and then compared PCNEM with our algorithm to these two. We showed that with as few as 64 features, our method successfully outperforms the other methods across all 3 noise levels. Next, we relaxed the transitive closure constraint and extended the simulation study to the entire DAG space to assess the performance as a function of off-target frequencies. So for this, we sampled networks from the highest and lowest scoring pathways in KEGG. Now, the histograms here show the off-target frequencies for these networks that were sampled from the highest and lowest scoring pathways. Again, we simulated data for 3 different pairs of error rates and 4 different number of phenotypic effects. And we showed that the performance of PCNEM was consistent and independent of the degree of off-target frequencies. So far, we assumed that we knew the error rates a priori, and we only focused on performance evaluation of network reconstruction. However, since, as I mentioned earlier, our algorithm can also estimate the error rates, we set up a study to assess the performance of network inference without a priori knowledge of the error rates. So for this, we sampled 30 DAGs with high off-target frequencies and generated data with 25 different pairs of error rates. The results are summarized here. On the x-axis, you have the true alpha value, and the box plots are colored according to the corresponding beta value used to generate the data. On the y-axis, you have performance again in terms of structural Hamming distance. The black solid line and dashed line correspond to performance of random guessing. Now, we showed that our method achieved really low structural Hamming distances, even for when one of the error rates were really high, and we consistently performed well for values as high as alpha equal to 0.5 and beta equal to 0.3, or vice versa. Now, this is important because we observe such noise level in our real data. So that brings us to our application. We applied PCNEM to Bartonella-Henselae infected RNAi screens. Now, Bartonella-Henselae is the causative agent of cat scratch disease. Romberg and Trutman et al. Identified a group of 8 genes in the focal adhesion complex to be involved in the uptake of this bacteria. The siRNAs designed to knock down these genes exhibited a high degree of off-target effects, making our model a good fit for inferring the relations between these genes. Now, the raw data consisted of single-cell image-based features, which we converted to binary gene-level data. So the final data set consisted of 288 features across 35 knockdown experiments. We applied PCNEM to the entire data set and 100 bootstrap samples with a threshold of 50% for support. And we noticed that almost all the edges that were inferred in the bootstrap graph provided support for a subset of the edges in the MLE estimate. The optimal PCNEM inferred a network that was in good accordance with knowledge from existing literature. And in addition to known interactions, we also identified novel interactions from CDC42 to TALEN1 and RAC1 to TALEN1. These have been reported in the string database. We believe that these interactions, subject to further experiments, could shed novel insights on the infection mechanism. That brings us close to the end of my talk. So in summary, we developed a novel extension of NEMS called probabilistic combinatorial nested effects model that explicitly accounts for combinatorial off-target effects in a probabilistic manner. It is this probabilistic aspect of the model that makes PCNEMs identifiable in the DAG space. We performed an exhaustive simulation study where we showed that PCNEM was invariantly robust or robustly invariant to the degree of off-target frequencies and high error rates. We also inferred a network on infection screening data that was in good accordance with knowledge from literature, and we inferred novel interactions. Our method has been implemented as a part of the existing NEM package and is available on our group GitHub in case you're interested. With that, I'd like to thank my professor, Nico Berenwinkel, senior scientist Jack Quippers, and a former colleague, Fabian Schmick. I'd also like to thank our collaborators at University of Basel. And last but definitely not the least, I'd like to thank you all for your attention. I'd be happy to take questions.
Questions for Sumana?
Hey, thanks for the great talk. So I'm coming from completely ignorant. I don't know any about this, but I'm wondering if the fact that, or how your matrices are set up, you are not, or you might be considering off targets when maybe the actual phenomenon that's happening is that the functional outcome that you're getting is actually disrupting a feedback loop onto the same kind of system. So I'm wondering if if the methodology takes that into account in any way.
Okay, so unfortunately, we can't deal with cycles yet. And the only way we can do this is with time series data. So this is a big problem that we have. But also, we're not inferring these perturbation maps. So these are based on seed sequences of the siRNAs that are designed. And targets can basically perform something like a blast and sees sequence similarities and give some kind of a score for how likely it is for that gene also to be targeted with that siRNA.
Thank you.
So I actually had a very similar question. So how did you deal with that when you did your validation with the KEK pathways? Did you limit yourself to only feed-forward pathways?
I didn't get it. So in the KEK pathways, yeah, we sampled only DAGs or transitively closed networks, basically. We didn't account for feedback loop, if that was your question.
Right. So the paths that you tried didn't have any feedback loops.
No.
Okay.
Maybe slightly orthogonal question. You, as I understand, you're modeling your likelihood of the observed data that you're considering. Assume that you can model the final phenotypic observations using a Gaussian noise. So is that
So you mean
always?
You mean like extended to continuous data, or?
Yeah, or other noise distributions that you could imagine, depending on what the outcomes were. Is it a framework general that you can plug in any observation model, or what are the limitations?
I think the likelihood function has to be tweaked, and that was definitely something that we have not included as of now. But the original NEMS already has this, where they kind of model continuous the data in a different way. And we didn't think about that, but definitely that's a way to go in terms of extending to continuous data.
Okay. So if there are no further questions, let's thanks, Sumana, again.